Okay, so good morning. Have a seat. You're the first student and you're also late. So we'll start with you. Okay? Right. So today we are going to be covering acute inflammation. So can you tell me what is your idea of inflammation? Redness. You think inflammation means red? Because whenever something gets inflamed, your skin and all becomes red. Is that the reason? Any other? Yeah, that's a good enough reason. That is actually correct. Any other idea? What is inflammation? We have done cell injury, cell necrosis, and other things. Um, did you come across a response to those things? Suppose you had cell injury. You studied cell injury. So, if a cell in the body gets injured, what will the body do? It will try to repair it or it will respond to it. So, the purpose of inflammation is to respond to something. So, yes, you are so bright and early. 3 minutes late. 4 minutes late, 5 minutes late. All of you, please come. We will start in just a moment. But if you're all catching the 8 o'clock train, you would all have missed it. More will come, or should I start? More are coming. So, how shall we give punishment to those who come late? Some punishment should be given. So, if you come 5 minutes late, we will give you 5 marks less than what you require for passing. 45 out of 100. That is the best response. Now, usually it is senseless to scold people who are not culprits when the culprits are not present. But in your case, you all three came late anyway, so you deserve some of this despite the fact that you actually arrived. So, what is the reason for the lateness? Any reason? You never had lectures in your first year in 8 to 9. That's the reason. You got used to 9 o'clock lectures. But that was not how it was in MAMC. I'm from Mamsi very long ago, 40, 30 years ago, 35 years ago, or even more than that. But the lectures used to start at 8, even for anatomy and physiology. Everything used to start at 8. Why has this not been done at AIMS? I don't know. But you can come in late. You know, come in, come in, come in, come in. So I think we need to tell the anatomists and the physiologists and the biochemists that if they want to train doctors, they also have to come at 8 o'clock and start teaching. Otherwise, this is the consequence. है So let us send a message to Tony. Told. So all your subsequent generations can tell that we identified the problem and we cured the problem. Hopefully. Unless they don't cure it. Okay. So nevertheless, let us start. I think at least somebody is here. The person who takes your attendance will take attendance without allowing proxy. Okay. So you go to their bench, take their attendance, and anybody who arrives after 10 past 8 gets to attend the lecture but doesn't get the attendance. go and take the attendance. Not now, at 10 minutes past 8. ठीक है, अभी 4 मिनट बच गए हैं. Okay, so we start with a lecture on acute inflammation. Acute inflammation is one of the most important processes which you all need to study in this pathology, because this is a recurring theme. You're going to have inflammatory diseases all through your life. Apart from malignancies, they are the most important disease which the human body gets, inflammation. So what is it? Let's look at the definitions first. Inflammation is a complex, non-specific, stereotyped protective reaction of vascularized tissue in response to pathogenic injury. Each of these words is important. Non-specific means regardless of injury, the reaction is same. Stereotype means regardless of what initiates inflammation, the same pattern follows throughout. The protective reaction, the purpose is protective, and it only happens in vascularized tissue. So vascularized tissue means the dead tissues of the body cannot have inflammation. You call it acute when it's a reaction of short duration. You call it chronic where it's a longer duration process of inflammation and repair occur at the same time. So this is the definition. Repair is a process by which the tissue which is lost by necrosis, which you studied earlier, or inflammation, which you're studying now, or both, is reconstituted and is derived from the Latin word inflammatio, set on fire. That's why you call it an inflammatory speech. So all kinds of injurious agents can set off inflammation. Hypoxia, which is studied as a cell injury cause, can cause inflammation. Physical agents, like for example, getting hit, a scratch, it could be irradiation, it could be barotrauma, all kinds of physical agents. Chemical agents, all kinds of corrosive substances and other chemicals which damage from inside the body by metabolic processes. All the infectious agents, which is extremely important, so bacterial, viral, parasites, microbial toxins. Immunological reaction, that means the body's own immune mechanism can cause inflammation by setting it off. A patient having genetic derangements can have abnormal inflammations, and even nutritional imbalance can cause inflammation. So almost anything which can affect the body can ultimately trigger inflammation, but it's supposed to be protective. Having said that, it can be potentially harmful, as you all must have seen in the COVID pandemic, that too much inflammation is not good for the body. The purpose is to destroy, dilute, or wall off the injurious agent. So the injury is there, and the body wants to limit the injury and wall off the agent which is causing the injury, and then repair what has been harmed. The purpose of inflammation is to bring serum proteins and neutrophils to the site of injury. So you want serum proteins which contains things like complement, which you have read about, and neutrophils which serve the function of inflammation to the site. So that is why you are having the inflammation. And the purpose of these cells and these proteins is to remove the injurious agent, either by diluting it, destroying it, or walling it off. Now, too much of inflammation is a cause of tissue injury. It means that the injurious, you're trying to injure the injuring agent back, like a microbe, but you end up injuring your own body. So these are the cardinal signs of acute inflammation. So you have calor, that is hot, rubor, that's red or blushing, tumor, that is growth or swelling, dolor, that is pain, and then functio lisa, which means functional impairment. So these were described way back in 330 BC by Celsus, and then this loss of function was added by Virchow in 1902. So why do you call it to set on fire? Because it becomes red and it's painful, so it's like fire. So you have redness, swelling, it becomes hot to touch, it sets off the pain endings, and you can't use it. Inflammation can be divided into acute, subacute, and chronic. Now, mostly clinicians cause any process as acute if it is less than 4 weeks. We call anything chronic if it is more than 6 weeks, and in between they call it subacute. That may or may not work for pathology. Pathology, we call it acute, chronic, and acute and chronic rather than subacute. And what you call acute inflammation typically is very rapid, that is seconds to minutes. So very short duration, minutes, hours, or a few days. Whereas chronic is slow, insidious, very long duration, month, like that. And the cells involved are different. Whenever anything is inflamed, you use certain standard terminologies. Pancreatitis, meningitis, pericarditis. So this itis is there. Anything, if you add itis, studentitis, you can call anything as inflammation. So what is this inflammatory process? You have an initiation, an amplification, and a termination, and it has certain components. There is a vascular dilatation, an extravasation of cells and proteins, and then the cellular functions are observed. Okay, so two of these that means are vascular events. So you have to, that is why we are using the word vascularized tissue, because if it's all about the blood vessels. So the vasodilatation and the leakage or the increased vascular permeability are vascular events, and the last only is the cellular event. So if you look at this, this is an overview of what all happens. Everything in inflammation is illustrated here. The vessels dilate, the cells come out, the plasma proteins comes out. Okay, so let us look at it stage by stage. The best response of an inflammation is, if somebody is angry with you and gives you a slap, what happens to the cheek? Slap somebody. Becomes, sometimes, the mark of 4 fingers can also come. Why is that? Anybody knows why that happens? You studied it in physiology. It's written also here. So, can you explain what happens? No, that is too simple. I've been talking about vasodilation for a long time now. Can you tell something better? What sets it off? Why do you think if I take a pointy pencil and give a scratch, one red line will come on your skin? What does the sharp pointy pencil do? Okay, no. So direct injury is not. Okay, what it does is that there are mast cells present in the skin, and physical stimulation causes the mast cells to degranulate. So almost all injury is initially started by the mast cells, and their function is only this to cause. What do mast cells contain? Hmm? They contain histamine. What does histamine do? So there you have the answer to almost 90% of inflammation. If you know this much also, it is enough. So when you took the sharp pointy pencil or you slap somebody for coming late, there will be a red mark, which is because the mast cells got stimulated. Subsequently, what else does histamine do? It, what is Lewis triple response? You must have studied in physiology. Red line, wheel, flare. No, sorry, red line, flare, wheel. First there is red line, then there is flare, and then there is a wheel. Initially, you might have studied the axon reflex also. When you take a sharp pointed object and you scratch the person, initially you get a white line. Why is that? That's called axon reflex. There is a reflex vasoconstriction which is instantaneous because it's mediated by nerves. Then the mast cells histamine comes, then it becomes red. Now, because histamine is a little soluble, it diffuses out and therefore causes a flare. That means the red line goes beyond the area where you stroked it. So in areas where mast cells were not initially stimulated also, histamine is flowing because it's in this tissue, and then it causes a wheel. Wheel means increased permeability. So fluid comes out of the blood in that area. So These are the, in 3 to 50 seconds, red line. Within a minute, there is a flash or flare, and in 5 minutes, that area becomes, you must have scratched yourself and so on, becomes very thick. So, this is all the purpose, delivering the plasma and cells. Main mediator is histamine, and this stasis, that means whenever there is vasodilatation like this, so laminar flow is there, now it is dilated. So there is slow flow. You know the Bernoulli principle, the more the diameter, the slower the fluid flows. So this enables these cells to start attaching to the blood vessels. So cells can also come out. So if you have an arteriole, capillaries and venule, there is dilatation here. Dilatation means increased flow through the capillaries. There is increased exudation of plasma protein and cells, and the veins also increase in diameter. That's also important. Now, normal vascular permeability retains all the proteins. That's why you have oncotic pressure. Now, if there is leakage of plasma-rich fluid because the increased vascular permeability, that means protein is going out. And if protein goes out, fluid also goes out along with it. And so that's why the swelling develops. Right? It is simple, your old Starling's mechanisms only. So there are two kinds of things which you need to understand what they are. One is a transidate. A transidate means that protein poor fluid is coming out anywhere in the body. Now a transidate can be something like edema, in which the oncotic pressure, the plasma proteins in the blood have reduced because he has Kwashiorkor, marasmus or some kind of thing, or the proteins are not being produced, so tissue comes out and water comes out. That's called a transidate. It is not associated with vascular permeability. It is an inability of the blood to hold the water. The blood cannot hold the water, so water comes out. Whereas an exudate is an active process. The vascular permeability increases. So we are just, let us see what this is. So this will be less than 0.015 and this will be more than 1.020. Now, why does this increased vascular permeability occur? So there are different kinds of responses: immediate transient, immediate sustained, delayed response. So let's see what all happens. Why all does vascular permeability increase? First and most important, the one which is caused by this mast cell and histamine, is contraction of endothelial cells. So I think there is a subsequent slide where you can see this is an immediate transient response. If you have endothelial cell contraction, the endothelial cell which was like this has contracted. So the gaps increase. The gap increases, that means there is nothing holding the fluid inside. So the fluid comes out, the plasma protein comes out. It's very simple. That's what histamine does. So typically there are some basement membranes and other things which still try to hold it together, but still it comes out. Okay? So loss of the protein from plasma leads to the edema. Wherever the protein goes, the fluid follows. So these endothelial gaps are what cause it. Now this kind of thing is called an immediate transient response. It's mediated by histamine, but there are other substances also which can produce chemical mediators in a subsequent lecture. And delayed type is also there. Now why does delayed type occur? So before we go to delayed type, let us look at this immediate sustained. Now we called it immediate transient. What does it mean? It contracted, after some time it will relax. When it relaxes, it will come back to position and the gaps close, and therefore no more exudation, no more increased vascular permeability. When will it be sustained? When the cell itself is gone. Okay, the cell has vanished for some reason. So what all can happen? You might have very severe injury which damaged the endothelial cell also. Yeah, so far we said the damage only stimulated the mast cell. Now the damage has killed the endothelial cell directly. That can happen in a burn. So it's called an endothelial cell necrosis. Or there could be some histolytic bacterial enzymes which are destroying it. Immediately it will start. and it will last well over 24 hours. So it will keep on happening. So when you get a burn, you'll keep on exuding because blood vessels are permanently damaged. And it only stops exuding when the endothelial cells are regenerated. The endothelium comes back, that's the only time that you can get it back. So, like this, you get blisters in the skin, all of you might have times experience. That's the same thing, a skin burn blister. Now, there is another type which is delayed but prolonged. Okay, so delayed means it doesn't start immediately, it starts after a few hours. And then having one started, it stays for a long time. Now there are two processes which are mostly involved in this. One is called endothelial retraction. This is a kind of cytoskeletal rearrangement and is mediated by certain inflammatory mediators like interferons, interleukin 1, TNF. So these are certain inflammatory mediators which set off a delayed but prolonged response by rearranging the cytoskeleton. So the endothelial cell has become more swollen and the gaps have increased. Right. So typically seen in hypoxia. The other thing is apoptosis. Apoptosis means that the endothelium, you know what is apoptosis? You had a lecture on it. So what happens if the endothelial cell undergoes apoptosis? It's not going to die immediately. It will take some time to die because the lethal injury has been developed, has been delivered, but it takes time for the cell to undergo the apoptosis. Having died, what happens? Until it is regenerated, not going to serve its function. Typically seen in ultraviolet rays in sunburn. Right. So there are many other mechanisms like transcytosis or pinocytosis increases. You can have leukocyte mediated injury to these. So one of the feature of leukocyte activation, newly formed vessels, whenever you have inflammation being reconstituted, they become very leaky, typically seen in granulation tissue which will come and in repair. So these are other causes where there is increased exudation of fluid for these reasons. That's pinocytosis. You can see that this is a lumen and this endothelial cell is full of pinocytotic vesicles. And you can see how this is the inside the vessel. They have injected a dye, and the dye is inside these vessels. So this is a graphical representation of the immediate transient, immediate sustained, delayed prolonged. Almost all these various causes will do one or the other of these. So is that clear? Do you have any questions on the basic inflammatory process? This is something which will recur throughout your studies everywhere and in your clinical practice. Important that you know it well. Do you have any questions or you like to discuss anything about it? Okay. If not, then let us look at certain important terms which you need to understand. One is edema. Edema is tissue swelling because of excess fluid anywhere. That means your arm might be edematous, your whole body might be edematous, your eyes may be edematous, your lungs may be edematous. So anywhere there is more fluid is called edema. Transudate we already discussed what it is, low protein content because of it's a transudate. It is because of reduced oncotic pressure. Whereas an exudate is fluid with high protein content and high cellular content. An effusion means collection of fluid in any body cavity, pleural, peritoneal, or any other cavity. Name any other cavity. Pericardium. What do you call a fluid in the pericardium? Pericardial effusion. Any other cavity you might know? You can have, what is a hydrocele? Have you heard of hydrocele? What is that? In the tunica vaginalis. So that is also derived from the peritoneal cavity only during embryogenesis. So everything is an effusion. And an effusion can be serous. Serous means clear, okay, or straw colored fluid, usually a transudate. Sanguinous, containing blood. Fibrinous means rich in fibrin. What's fibrin? You have studied clotting. So if you are exuding protein into a fluid and it clots, it will form fibrin. Sometimes it can form clots on the surface of whatever is lining, pericardial surface, pleural surface. That will be called as a fibrinous exudate. Purulent means pus. What is pus? Pus. So pus is basically liquefactive necrosis. It is fluidized, inflamed necrotic tissue. So mostly composed of dead cells, neutrophils, dying neutrophils, and it is liquefied because of the action of the neutrophil enzymes. Suppurative means it's a process. You describe a process as productive of purulent exudates. So pus is the purulent material, so commonly which you use. Now let's go on to see how the leukocytes come out. So the vascular events are over, and now we come to the leukocyte events or WBC. The important function because the leukocytes have to go to the injury to mount the host defense. What is the host defense? Ingesting organism is ingested, the offending organism rather, kills the bacteria, kills the microbes, eats the necrotic tissue and takes it away in the blood. The neutrophil comes out of the blood, eats whatever it wants, goes back into the blood and the blood carries it away. But where will it go from blood? Ultimately, it will digest this substance and release its constituent enzymes. You will get urea and creatinine at the end of whatever thing is being removed. है ना? So, double-edged weapon. What does that mean? It means that tissue damage is essential to the inflammatory process. You are trying to digest what you don't want and you end up digesting what you want also. Yeah, leukocyte products that destroy microbes and necrotic tissue can also injure normal hosts. So this is how things happen. Blood flow slows down, cells stick to the surface and they squeeze out. See, there is this process. So what are the things? So normally there is laminar flow. Then there is margination and rolling. Margination means they come to the periphery. Rolling means they stick but not that hard. The stickiness which is meant to slow them but not stop them. Once they come to a standstill, then they undergo adhesion. These are stronger receptors. Once they are attached to the receptors, they actively using actin pseudopodia squeeze out and come out like this. Right. So you can see in a tissue section how margination has occurred. That this is a dilated vessel. All the WBCs are sticky. Same thing, they have put a color, they have made the phagocyte eat something. How all the neutrophils are marginating. The diagrammatic representation showing the various processes of what we just discussed. So you can see two kinds of receptors are there. How early or late are you for the next class? Next class क्या है आपका? क्या बोला इसने? CCM. You're too early for CCM class. है ना? It's only starting at 9 o'clock. If you want to attend the class, please come in. So what do you think this is? Somebody has drawn a diagram. This is a lecture I took maybe 20 years ago, but I still tried because I like it and haven't done. What do you think is happening here? This is called a Boyden's chamber. So Boyden's chamber uses a membrane which has pores through which the WBCs can squeeze through it. So you take a Buffy coat and you put it on one side, and on the other side you put the agent which you want to test and see whether the WBC will squeeze through and come out or not. That's how they discovered all this. chemotactic agents. So what are the things which make WBCs attached to the vessels? These are all the main groups. You have integrins, selectins, the immunoglobulin gene family, of which integrins are a part, and then you have glycoproteins or mucin-like substances. So let's look at selectins. This you have to remember. There are three kinds of selectin: L, E, and P. And the easiest way to remember is L is lymphocyte selectin, that means or lymphocyte. E means it's expressed on the endothelial cell, which is also called, in the old days we used to call it ELAM1. We had all kinds of names, so easier to remember. Now they make it more difficult by giving the same standard terminology. Then P. Now the way to remember P is not just that it's in platelet, but it's also in pallid or Weibel pallid bodies. That means it is also present in endothelial cells. Okay? So sometimes people get confused and think that P-selectin is only for platelets. That's not true. Then you have integrins, which are a very large group. We used to call them in the old days VLA and LFA1, but now you just have all kinds of names. What is the function of these selectins? These are calcium dependent attachments which has a fast on, fast off kind of attachment. That means that it will attach, but the attachment is not very strong and it will give way. And low affinity. That means that the attachment is not strong. So obviously this describes the kind of attachment you require for rolling. Exactly. So rolling. The immunoglobulin family molecules like ACAM1, VCAM1 and PCAM, they are stronger attachments and are the most definitive for going through. So you can remember this: leukocytes attach to endothelium by these methods. So P-selectin and Cl binds to sialyl Lewis X and causes rolling. E-selectin, again rolling and adhesion. Then ICAM1, strong adhesion. PCAM1, addition of more cell types than just the ICAM1. Then glycam1 binds to L-selectin of the leukocyte, which is important for homing. Now, what is homing? Do you understand what is homing? Homing means just like homing pigeon goes back to its precise house, similarly certain cells go to certain precise locations. So if it belongs in a lymph node, it will home to the lymph node. And it achieves this because of these kinds of L-selectin glycam1 type of expression. So only the endothelium in that area has a adhesion molecule and not vessels elsewhere. Okay? And then migration. So migration through endothelial gaps is through another substance called CD31. We used to call PCAM. So how does leukocyte adhesion takes place? One is the immediate. Immediate, it has to be preformed. So something which is preformed. So Weibel pellet bodies are small granule-like things which contain the P-selectin, and whenever there is inflammation and a mediator binds to the endothelium, they get activated and they release this P-selectin. And so the endothelium becomes sticky. When the WBC comes, it will stick there. So typically stimulated by histamine, thrombin and platelet activity factor. One is redistribution, the other is induction. Induction means expression of the adhesion molecule. This is slow process. So it will take 1 or 2 hours and the cell will produce these like TNF, IL-1, chemokines. These are produced by the endothelium when it is activated. And then the classic expression is VCAM1 and ICAM1 or immunoglobulin gene family molecules, which cause increased avidity of binding. Okay, so diaperesis or squeezing through is because of PCAM1. Retention in the soft tissue, in the extracellular area is by CD44. And there are chemotactic agents which make them move towards other places, like we talked about in the Boyden chamber. So this is an illustration of whatever I told you. They come, they stick. E-selectin and P-selectin are there, they stick, right? And then stronger sticking because ICAM1 and all have started attaching. Once it is strongly stuck, then PCAM1 will make it squeeze through, or CD31, and it's squeezing through. Now it will move because of the chemokine. Suppose there's a chemokine here, it will move because for having arrived there, they will do their function. That means they will eat the microbe, for example. So, and having finished their function, they'll go back. Not that they're going to stay there forever. So they have to go back to the blood vessel and then they have to vanish. This is just another representation of the same thing. Now we come to what inflammation looks like, and these are slides which you will get. This is, what do you think this tissue is? Anybody? Got kind of fiber-like things. There are certain lines over here which you can see. Some lines. Cardiac muscle. Brilliant. Full marks to all of those who thought of this. Yes, this is cardiac muscle. But what's wrong with this cardiac muscle? Yes, there are a lot of neutrophils. Too many neutrophils in between the cells. It means what has happened? This cardiac muscle has become inflamed. It has achieved some injury, for example, a heart attack. It had a myocardial infarction, let's say. And at the edge where blood is still flowing, because it requires vascularized tissue, the area will undergo inflammation. And you must have studied that in necrosis, that necrosis elicits inflammation. So that's what has happened. So these three dots, wherever you see three dots, it's neutrophil. Two dots is eosinophil. That means there are two nuclear lobes. One dot is lymphocyte. Usually, I mean, easiest way to remember when you're seeing under low power. So if you are seeing too many dots, that means all these neutrophils are the nuclei. Okay, what's happening over here? Congested blood vessel. So the blood vessel has become congested. Okay, so this is how it comes. Immediately you get an edema. After that, the neutrophils come, and last to come are the monocytes and the macrophages, because they express different kinds of receptors and different kinds of mediators, both at the endothelial level and at the monocyte level. So by changing what the endothelial cell expresses over a period of time, different types of cells will come their function. How did I say that it's a congested vessel? Because it is having a lumen which is fairly large. Now, normally, whenever you take sections from any tissue, the blood vessels don't have too much blood inside them. So, when you look at lots of sections, you will realize that blood vessels are rarely full of blood. So, whenever you find a blood vessel which has too much blood, it is a sign that something is congested. For example, we typically get it in, like, for example, the GI tract. So in the GI tract, frequently what happens is that there is a gangrenous bowel or there is an obstruction. They tie it, so the actual injury can be by the operation itself. So because you have tied it, that area will get congested. So some blood vessels on the outside serosa will have too much blood, but rarely you will find blood inside. There is blood inside where you don't expect the vessels to be congested by the tying operation and all. Then you suspect that there is some other active process going on, because of which the vessel itself has dilated and therefore it is congested. Plus we also get an estimate by experience at how big are the vessels which you normally expect to be in a particular tissue, and based on that you decide. So there are other adhesion molecules like adenosines which are specific for homing, and CD44, which is important, which is an extremely important molecule which has numerous functions, one of which is T binding to the endothelium, or for example coming out in high endothelial venules. So there are two processes. One is chemotaxis, which is moving along a gradient, and another is haptotaxis, which is a response to chemotactic agent is bound to a matrix. That means there is a gradient in the matrix itself. The gradient cannot change because the chemotactic agent is immobilized on the matrix. So what is chemotaxis? It can be because of exogenous agents or endogenous agents. So exogenous agents are typically bacterial products, and endogenous is typically the complement or leukotrienes. So we will do chemical mediators soon. So leukocytes which are coming out in the site, they require activation, and this activation is typically because of arachidonic acid metabolites, and the result of this activation is degranulation and secretion of lysosomal enzymes. So there are two ways in which a neutrophil can function. It can eat something and then kill it, or it can directly degranulate outside and kill something outside. So if it degranulates, typically it does degranulate. It is non-specific. It will kill whatever is in the vicinity, including host tissue. So what are the activation markers? Most important are the toll-like receptors. You must have read about TLRs when you were reading about COVID, or you might have done it in biochemistry. So TLRs are the pattern recognition receptors, and they get activated, and when they are, then the leukocytes get activated. The other way is by binding of cytokines, and usually this is by the transmembrane G protein coupled receptors. So these receptors can recognize these N-formyl methionine, chemokines, lipid mediators. So number of things can be recognized by the transmembrane receptors. So you can see these diagrammatically represented. This is from your Robbins only. So these transprotein G protein receptors, they can recognize these chemokines and other mediators. And then cytoskeletal changes, calcium changes, cytoskeletal changes, migration. Microbes directly recognized by TLRs with co-stimulation, and then there is leukocyte activation, which causes killing. And other receptors like mannose receptors will recognize the mannose, and then they will again get activated with killing of microbes. Sometimes you can get an adhesion deficiency. What is this? So each of the numerous list of proteins and other molecules which we described, they can be deficient. है ना? It's like sometimes attention can be deficient. देखिए मैम, है ना? So this is an example of what happens when you put it in a Boyden chamber. This is the pseudopodia. This is in cell culture. Take a cell culture flask. You put a chemotactic agent. You can see how the pseudopodia are coming out. So what kind of microscope you will see to visualize this? You see an inverted microscope. Don't kill the cell. You want to see the living cell. So cell culture, you put on an inverted microscope and use phase contrast. So you know what is phase contrast? Phase contrast means you increase the contrast, or you put a phase disk so that parallel light which is little far away is going through, that bends the light in such a way that even unstained tissue is able to be visualized. So you can clearly observe living cells and putting out pseudopodia and moving in one direction. You can evaluate all this. Now in biochemistry, you have gone through all these pathways. Now what do you have to remember? All you have to remember is that almost all of these pathways are used as receptors by one or the other chemical mediator. Second thing you can remember is that the same receptor can have more than one way in which it is coupled. That means it binds to one receptor and does function 1. It binds to receptor 2 and does function 2 also. So by having a variation in which all receptors can get activated in which all different cell types, the same thing can have a function in endothelial cell, another function in neutrophil, third function in lymphocyte. So it's impossible for anybody to remember all that. That's not the function. You just know that these are the basic ways. The IP3, the calcium, the protein kinase C, the tyrosine kinase, protein phosphorylation, dephosphorylation, cyclic AMP, almost all of these can be used. Yeah, but the most important of these are the G protein. They have given one example here. Okay? Actin ultimate end product is microfilament contraction. So it can bind to other receptors coupled differently. The functions are phagocytosis, breakdown, oxidative killing. What is phagocytosis? The active process. Pattern recognition receptors can trigger it. Opsonins. Opsonins are things that make something tasty. So C3b receptors and immunoglobulin Fc receptors. So what is an Fc receptor? Antibody binds to the microbe, attaches to the phagocyte because the Fc receptor binds to it, and then the phagocyte will eat whatever it is which has been bound by the antibody. Same thing with complement. Anything coated by C3b will attach to C3 receptor, internalized, and then whatever is inside is hydrolyzed. There is an engulfment because of pseudopod formation and phagosome formation. Then lysosomes fuse with it to produce a phagolysosome. Right, this phagolysosome kills whatever is. It activates all the hydrolases which are kept in an inactive form. So the lysosome walls off the hydrolase from the rest of the cell, but if it fuses with the phagosome, then the hydrolase is released into the phagosome. So it eats it, digests, the indigestible materials come out and they get carried away in the blood. What happens if they get carried away in the blood? They still have to exit the blood in some way. Can you name things which cannot be removed at the end? You might have done it in your cell engineering. Almost everything can be broken down in the body except few things. Anyone? Carbon. So carbon, nothing can do about it. It's there means it is there for life. Delhi pollution or those of you who smoke. Then somebody said something else. Silica. Okay, if you go and breathe, you're a miner and you breathe that. Any other substance? What about normal things? Breakdown of this. One of the breakdown products is called lipofuscin. Lipofuscin is a lipidaceous kind of a material which has cross-linked lipids and which are impossible to break down any further. So they stay around. The only way to eliminate lipofuscin is if it is in a lumen, for example, GI tract. In the GI tract, the cell gets shed. Cell gets shed, it goes away and takes the lipofuscin with it. Otherwise, you can't eliminate it. So this is more representation of the same killing. So how do microbes get killed? Hmm? You know about the function of this? Another. Yeah, what is this? You have all studied the function of NAD and FAD, NADP, FADP. They are very important in mitochondria, but they are also important in the granules of the neutrophil. You have this NADPH oxidase in the granule of the leukocyte, whose function is to generate oxidative burst or oxygen free radicals. They generate this free radical, and that free radical is what kills it. So there will be subsequent slides which will explain it in slightly more detail. These are also called reactive oxygen species or ROS. You studied that in your cell engineering, I'm sure. So reactive oxygen species are generated for killing microbes. So if there's a bacterium, this NADPH is what is most important. It generates the O2 minus free radical. But to potentiate this O2 minus, you can also use this MPO or myeloperoxidase to create something which is more potent at killing the bacteria. Okay, so MPO is not essential, but this NADPH is essential. This can only function if this is already there. If this is not there, this cannot function. So this is how the phagocytes look like. You have the neutrophil, monocyte, lymphocyte, neutrophils, eosinophil, two lobes and red granules, basophil, which has basophilic granules, Then you have the mast cells, the macrophages. So phagocytosis are two main types of killing: oxygen dependent and independent. Independent is major basic protein seen in eosinophils. Then lysozyme, which is kind of effective for cell wall. Lactoferrin, which is binds to iron and prevents bacteria from using it, so the bacteria die. Defensins, proteins which bind to specific species of fungi or other things and inactivate them. See, so even fungi produce defensins, seen in invertebrates also. So neutrophils have all these granules. You have the primary granule, okay, then you have the secondary granule or the specific granule and the C particle. So let's see how it works. The oxygen dependent killing creates a superoxide, which by superoxide dismutase becomes H2O2, which after catalase becomes inactivated, or by myeloperoxidase becomes HOCl, which is also antimicrobial. Iron is required for many of these activities. So respiratory burst means that the amount of chemiluminescence which is produced increases instead of like this. So if you have normal cells like this, those which are undergoing respiratory bursts are using much more oxygen. Okay, so these are the enzymes which are involved. Oxygen dependent mechanisms is went over. They cause the respiratory. Okay. Then they have the independent killing, which is the MPO, elastase, cathepsin, eosinophils, a major basic basophils. So these are all other substances like hydrolases, protease, lipase, cathepsin G. You have a BPI, which is bactericidal permeability increasing protein, defensins. So these are large in quantity, low potency. They are not that good at being effective, but they are also important. Lactoferrin binds again to the iron. Lysozyme degrades the cell wall. Tertiary granules are C particles. They allow the neutrophil to digest the extravascular compartment as they move through it. So these are all the important functions which you might have done earlier in types of cell death. So neutrophil extracellular traps. One of the things the neutrophil does is that it commits, like throwing yourself on the bomb. It throws itself on the offending agent, kills itself in the process, and the nucleus of this gets extruded out in a complex way which you might have studied to form an extracellular fibrillar network. This fibrillar network, all the antimicrobials get attached and subserve their function. So the microbes are inhibited by spread by getting trapped by this fibril, and the bactericidal proteins, the defensins, the BPI, the complements, the immunoglobulins, all of them will immobilize and destroy the microbe while this is. So this is extremely important, and we discovered this not very long ago. When I was doing undergraduation, we didn't even know that there are things called neutrophil extracellular traps. This is important that azurophils are the earliest granules to come. Specific granules come at the myelocyte stage. When you look, you have studied promyelocyte, myelocyte, metamyelocyte. So specific granules come at myelocyte stage. So to summarize, the leukocyte activation mostly is important for uh, act, uh, important for getting the leukocyte functional, usually by external or other chemical mediators. Function is increase arachidonic acid metabolism, degranulation, cytokine adhesion production, phagocytosis. So what do you think is happening here? Blood vessel. This is what kind of tissue? There's nothing here. So what do you what does it mean when there's nothing there? Full of fluid, or it's edematous. So this is edema fluid. And what are these cells which are everywhere? Leukocytes. And what is this cell which is here? Little long. So this is a fibroblast. So this is edematous tissue with lot of inflammatory. Okay, so who can recognize this cell? Anybody can recognize this cell? So this is a plasma cell. It's a round thing and it is a perinuclear half or a clear area over there. What do you think are, okay, let's ask this one. That's probably a lymphocyte, all and round. What are these cells? So these are some other host cells, some tissue which is getting inflamed, no? Epithelial cells are all. And this is, what do you think this is? Entire thing. Okay, here we have some normal tissue. What has happened to normal tissue here? Been replaced by neutrophils. This is liquefactive necrosis of that tissue. This meninges. What is this substance? Pus. So somebody, that's why the patient died, because he had so much of inflammation. Much inflammation, nobody can sustain. So this is a colon section, and what do you think are these? With yellow, yellow normally means, okay, one is pus, one is fat, another one necrosis. So necrosis also is yellow in color. So this is necrotic material, but this necrotic material is stuck to the surface, and so you, because it is stuck, you can't remove it. They call it a pseudomembrane. So that basically means necrotic stuff stuck to the wall of the intestine. The heart, and what's happened to the pericardium here? It's tiny, really dull. Here it is not just dull, it's actually having some stringy things which are coming out. So what do you think the stringy thing is? What is fibrin? Another word for clot. So normally you think of clot in the blood, where blood cells are trapped inside it. But you can have clot or a coagulum outside the blood also. You have acidic fluid, you can have a clot inside that. So those will be just like cobwebs or things which are floating, and they are full of fibrin strands. So this is heart muscle. It's also heart muscle. What has happened on the surface? Really greenish, almost yellowish, greenish material on the surface. So for some reason, fibrinous pericarditis is also called bread and butter pericarditis, because when you spread butter on bread, it looks like that. When you pull it apart, it gets a peculiar stringy thing, no? So they call it like that. So this is fibrinous pleuritis. Again, it is dull, and you have some strands which are coming out. Now, I said itis means inflammation. Some things are exception. Pneumonia is one. Do you use the word pneumonitis? You do. You will find in clinical practice there is a pneumonitis. People will use. You can use, but generally radiologists use that. So radiologists typically use the word consolidation in pneumonia for some peculiar reason. What's happened to this lung? What's this red over here? Blood. Red means blood. Why blood has come out? Because the vessels got dilated and the walls got damaged, so the blood came out. What is this blue areas? Means what is more? Some cells are more. Which cell it is likely to be? Neutrophils, inflammatory cells. So it is full of inflammatory cells. And if you go down under higher power, you can see these are the valvular septate, congested vessels. And instead of empty lumen, it's full of neutrophils. Here also you can see it's full of other inflammatory cells. Fibrinous serositis. So again there is dilated vessels edema here, fibrin deposition on the surface. Again serositis, thick inflammatory. The normal serosa is very thin, little loose areolar tissue and then a thin lining over here. Instead of that, it's become thick, full of cells as deposit. So you are having certain terminologies: serous, fibrinous, suppurative, ulcerative. You understand all this, right? They're just English. A serious inflammation. So fibrinous means there is a fibrin meshwork on this. Suppurative means productive of pus. So this has pus in the substance. We have already seen. Ulcerative means there is an ulcer which is produced. Ulcer is a breach in the surface of a mucosa. Erosion is a breach in the lining epithelium. Mucosa is a thicker structure than just the lining epithelium. So the end result of this is ultimately healing fibrosis. That's a subject for another class. So acute and chronic. Acute, you have ARDS, which you can get along with your COVID also. Acute transplant rejection, asthma, glomerulonephritis, reperfusion in shock. Chronic, we have chronic arthritis. You can have chronic asthma also. Even atherosclerosis is an inflammatory disease. Chronic rejection, chronic lung disease, all these can happen. So there is a termination. How does inflammation end? Neutrophils have a short lifespan. The cytokines vanish, they are washed away. And certain cytokines are anti-inflammatory. A new type of cytokine is produced which ends the inflammation. Typical examples are transforming growth factor beta and interleukin 10. So IL-10 you can remember is against everything which is pro-inflammatory. Want to kill inflammation, you put IL-10 in it. Then you can have anti-inflammatory metabolites like instead of lipoxygenase, instead of prostaglandins, you produce lipoxins, or you have proteins like resolvins. So that's how inflammation ends. And there are certain systemic effects which we will study in the chemical medium: fever, leukocytosis, and alpha. So I think it's time for your next class. Since you came 10 minutes late, we will have to end at this point. We will meet you again on the next class, which is tomorrow, or rather day after tomorrow. Okay, any questions on this? Okay, if there are no questions, he has already collected the attendance. Those who came in late, you got the lecture. So you should be happy that you got the lecture. What you cannot get? Thank you all.